Hi everyone, this is the IGCSE Human Biology past paper. This is from November 2023 and this is paper 1. So this paper takes about 1 hour and 45 minutes and it'll test you on the entire syllabus, and this paper is out of 90 marks. All right, let's start off with the first question that we have here. All right, question 1. The table gives components of the blood and their features. Complete the table by putting a tick in the box if the blood component shows the feature. All right, so here we have a bunch of features. There are 5 that we see over here. Let's start off with the red blood cell. So the red blood cell is responsible for transporting oxygen. So that's this tick here. And how it transports oxygen is that the oxygen gets carried by a protein called hemoglobin. All right, red blood cell is not a fluid because it's a cell and doesn't produce antibodies and doesn't engulf pathogens either. Let's go on to the next one, which is phagocyte. So phagocytes don't transport oxygen, they don't contain hemoglobin, they're not a fluid, they're a cell. So they're a type of white blood cell. So these two are white blood cells. And phagocytes are the ones that engulf pathogens. And you can see it from the name as well. So we talk about engulfing pathogen, that process is called phagocytosis. Right? And phagocytes undergo phagocytosis. Lymphocytes, the way they eliminate pathogens is by producing antibodies. So I've just included a diagram of an antibody. You can see that antibodies bind specifically to antigens, and antigens are found in all pathogens, in all cells, all viruses, and it is what helps the immune system recognize what belongs to the body and what is foreign. So antibodies would bind specifically to an antigen that would be foreign. Right. All right, let's move on to platelets. So platelets actually don't fit any of the description that's here. Platelets are responsible for blood clotting. And we'll go over that in the next question. Plasma is a fluid. So plasma is the fluid that makes up most of our blood. And our plasma is responsible for transporting things like carbon dioxide, glucose, amino acids, hormones, and heat throughout our entire body. All right, let's go on to part B. Explain why it is important that blood clots when the skin is open up or it is cut open. So I've included a diagram here to show you how it works. So platelets, and you can see the yellow parts there, get activated when there is a cut in the blood vessel. And what happens is that it converts this soluble fibrinogen into insoluble fibrin. And you can see that there. So this fibrin sort of forms a mesh, and that would prevent blood from leaving the blood vessels. And it would also prevent pathogens from entering the blood vessels as well. Another thing it also does is protects our skin when it's undergoing that healing process. All right, let me just remove this and let's write this down. All right, so we can say during blood clotting, a insoluble mesh is formed. This prevents blood from leaving the blood vessels and also prevents pathogens from entering the vessels, therefore preventing us against, we can say, infections or diseases. All right, let's go over the marking scheme. Number 1 forms a mesh. Number 2 prevents blood loss. Number 3 prevents entry of pathogens. And number 4, and that would protect us against infections. All right, let's go on to question 2. All right, to find out if a person has diabetes, they can be given a glucose tolerance test. This is how the test is done. The person does not eat for 12 hours. After 12 hours, blood glucose levels are measured. The person is then given a solution of glucose to drink. Then the blood glucose levels of the person are measured every 30 minutes for 30 hours. And then on the left, we have a person without diabetes, and here we have a person with diabetes. All right, so let's plot this on a graph. All right, so the x-axis over there would be the time in minutes. So let me write that down. So we can say time in minutes. And this goes up to about 180 minutes. All right, so we can have this be like this: 0, 30 minutes, 60 minutes, 90 minutes, 120, 150, 180. This would be 210 and 240. So you have to make sure that it goes up by the same interval. All right, the y-axis is going to be the blood glucose level in milligrams per 100 centimeter cube. Okay, I've zoomed it in a bit. Blood glucose levels, and it's in milligram, what was it, per 100 cm cube. Per 100 cm cube. All right. Now, the highest number that we have over here is 230. So let's try and fit that in the graph. I think we can go up in 50s. So this will be 0. This is 50. I'm just going to draw this a little closer. This is 100. This is 150, 200, 250, and 300. Okay, so even though there's extra space there, so long as we take up more than half of the grid space, and that's okay. All right, so I've included a little table over here so I can see while I'm plotting the graph. All right, so for a person without diabetes at 0 minutes, it is 65. So that would be here. At 30 minutes, it's 120. And then here, it's 130. And then at 90, it's 95. Okay, so it drops. At 120 minutes, it's 80. So that's at 150, it is 65. Okay, back to 65. All right. So that is a person without diabetes. And you can see here it says, join the points with straight lines. Okay, and I'm going to write this. Person without diabetes. All right, the next part is person with diabetes. So, whoa, at 0 minutes, it's 110. So it's over here. Then we have 215. and then 230, which is the highest. Then we have 200. Then we have 190. Then that's 180. And then 170. All right, I'm going to connect the points with straight lines. And then label that person with diabetes. Okay. Determine the difference between the highest and the lowest blood glucose levels for each person. All right, so for a person without diabetes, the highest would be, actually we can look at this here. Person without diabetes, the highest is 130, the lowest is 65. So the difference between those two is 65, because 130 minus 65 is 65. Now with diabetes, you've got 230, and the lowest is 110. So that is a difference of 120. Okay, so let's write that down. So it's 65 and 120. We can just say with diabetes, it was 130 minus 65. And then without, it was 230 minus 110. That was 120. All right. Let's move on to part 3. Give 3 differences between the glucose levels for the 2 people during the 3 hours. So if we look at the difference here, we can see that a person with diabetes has a higher blood glucose level, right, than the person without diabetes. Okay. Person with diabetes has a higher starting blood glucose level. Another thing we can also say is the difference, right? So a person without diabetes has a smaller range, whereas a person with diabetes has a much larger range between the lowest and highest blood glucose level. So we can say the person with diabetes has a larger difference between the largest and smallest value. All right. Okay, let's scroll up again. Okay, so you can see that with a person, for the person without diabetes, they started here and they ended here at 150 minutes. Whereas for the person with diabetes, they started here, but they still have not reached their baseline blood glucose that they did like they had in the beginning of the experiment. So we can say that the person with diabetes takes less time for their blood glucose to return to their baseline. All right. Part B, one symptom of diabetes is glucose in the urine. Describe how a sample of urine could be tested safely for glucose. So to test for the presence of glucose, we would undergo the Benedict's test. So the Benedict's test starts off as blue, and if there's a lot of sugar, then it'll have a red precipitate. And remember, I'm just going to draw a water bath here. So you would need to have a Bunsen burner, and you would put the test tube, that's supposed to be a test tube, in the water bath and leave it there for, let's say, 5 to 10 minutes. All right, let me just erase this. So first, add a sample of urine to a test tube, then add the Benedict's solution. You can say solution or you can say reagent, it's the same thing. Then place the test tube in a hot water bath for 10 minutes. You don't have to specify how long, I'm just writing how long it takes. All right, so one thing to note is that when you're dealing with a hot water bath, it's important that we wear goggles. If the color goes from blue to a red precipitate, then glucose is present. Now for the 5th mark, you'd have to mention a safety protocol. So for this, you have to make sure that you wear goggles because Benedict's solution is an irritant. So it's important that we don't get that in our skin or on our eyes. Make sure to wear safety goggles. All right, let's go over the marks. The first mark is putting the sample into the test tube. The second mark is to add the Benedict's solution. The third mark is to heat it in a water bath. The fourth mark is to wear goggles. And the 5th mark would be if it goes from blue to red, then glucose is present. All right, let's go on to the next question. Question 3. The table shows the flow of blood to different organs in a person at rest and during exercise. Okay, and then part A1, it says give the ratio of the blood flow to the heart when at rest and during exercise. So heart, rest, comparing it to during exercise. So these are the two numbers that we're looking at here. This 750, and now we have to divide this, both sides, by 250. So we get a 1 here, and then if we type 750 divided by 250, that gives us 3. So the ratio is 1 to 3. All right, the next part, calculate the percentage increase in the total flow of blood between rest and exercise. Give your answer in 2 significant figures. Okay, so if it's asking for total flow, we're looking at these two numbers here. So this is at rest and this is during exercise, and we divide that by 5,600. This is what we do. We find the difference. So the difference is this minus 5,000, what was it? 5,600. And then we divide it by 5,600 because that's the original value. And then we times that by 100 to get a percentage. So let's type that in the calculator. So 17500 divided by minus 5600, and then we divide that by 5600, and we get 2.125, and then we times that by 100, we should get 202.5%. But because it's asking for two significant figures, you would write that as 210%. So 210% increase. All right, let's move on to part 3. Explain why the total blood flow at rest is greater than the flow to the heart, skeletal muscle, skin, gut, and brain added together. Okay, let's scroll back up. So I'm going to add these numbers up together on the calculator. So 250 plus 1,000 plus 400 plus 1,400 plus 750 gives us, this whole thing gives us 3,800. And 3,800 is less than 5,600. So where is the rest of the blood flowing to? Well, it's probably flowing to other organs. So things like the lungs, for example, because the lungs is not on the list, or things like the liver or the kidneys, right? And that's because these organs are in need of oxygen and glucose from the blood, for example. All right, let's write that down. This is because blood is flowing to other parts of the body, such as the kidneys or lungs, as these organs need substances like oxygen from the blood. Okay, let's go over the marking scheme. So you could say other parts is 1 mark. The other mark is, you can give an example. So they said kidneys or lungs or liver, and they can also say arteries or veins. And that's because these organs need blood to be supplied there. So I said these organs may need substances like oxygen from the blood. Okay, describe the main changes between the blood flow values at rest and during exercise. So this question is asking us to describe. So what do we see between the rest and exercise values? Let's just erase this. So some of the major changes that stood out is, number 1 here, the total blood flow, right? It's almost around like 3 times as much of an increase. Another one is you would expect your skeletal muscles to also drastically increase as well, because we want more blood to our muscles so that they can undergo respiration. There's also an increase towards our skin and also to our heart as well. Although the jump in the increase in our heart is not as large as compared to the skeletal muscles or the skin or the total blood flow, which is why it's technically not mentioned in the marking scheme. Another thing as well is the gut. So here, the gut, there's actually a decrease. Okay, let's write that down. There is a large increase in the total blood flow to the skeletal muscles to the skin and to the heart is a large decrease in blood flow to the gut. All right, part 2, explain the change in blood flow rate to the skin when at rest and during exercise. So as we saw in this table earlier, there is an increase in blood flow towards the skin when exercising. And that's because when we're exercising, we're producing a lot more heat. And the reason why we're producing a lot more heat is because our muscles are undergoing more respiration, and respiration is a process that produces heat. And when we have more heat in our body, we need to get rid of that heat because we don't want our body temperature to be too high. One of the ways in which we decrease our body temperature is we undergo vasodilation. Let me just show a diagram of that. So here we can see when our body is warmer, there's a lot more blood flow to the surface of our skin. And what that happens, we have a lot more heat loss by radiation. That's why when we exercise, our skin tends to get redder, and that's because we have more blood flow towards the surface. All right, let me write that out. When we exercise, undergo more respiration, which is a process that produces heat. It's also the same reason why when we're cold, we shiver. And shivering is basically our skeletal muscles contracting in order for it to produce more heat. Reason why the blood flow to the skin increases is so that there will be more blood to the surface of the skin and therefore more heat loss via radiation. All right, let's go over the marks. So heat is generated during exercise. We exercise because of more respiration. That's actually 2 marks. And then therefore there's more blood to the skin. That's a 3rd mark. And heat loss via radiation. All right, let's go and do question 4. All right, question 4. The mass of DNA in a body cell changes during the cell cycle. Part A1, which structure in a cell contains DNA? So this would be chromosome. So let me just show you what a chromosome looks like. This is just some extra information. So here is a diagram. Right, so on the left here we have our DNA, and our DNA, because it's super, super, super long, it has to be nicely wrapped and nicely packaged into structures that we refer to as chromosomes. All right, in part 2, name one other type of nucleic acid. That would be the RNA. So RNA. You can also say mRNA or tRNA, that's fine. Part 3, give 3 differences between DNA and nucleic acid and the nucleic acid that you have named in Part A2. So basically the differences between RNA and DNA. So I'm just going to write RNA over here just for my own reference. So one of the main differences that we know is that DNA is double-stranded, right? So it kind of looks like this. It's a very bad drawing. So it's double-stranded, whereas RNA is single-stranded. Another difference is what sugar that they use. So I've included a diagram here. This is the DNA nucleotide, so you can see that there. This is the RNA nucleotide, you can see that there. So you can see the sugar is different because DNA we're using deoxyribose. And that's what the D in DNA stands for. DNA stands for deoxyribose nucleic acid. And the R in RNA stands for ribose, right? So that's one difference. Another difference is the bases, right? Do you see how for DNA you have A, C, G, and T? T is thymine. But for RNA, instead of a thymine, we have a uracil. So that is the 2 other differences. You can say has deoxyribose, has ribose, and then the third one is has thymine and has uracil. All right, let's move on to part B. The graph shows the changes in the mass of DNA in a body cell during mitosis. So here we can see that the mass starts off as 2, and then it doubles. So it goes from 2 to 4, and then all of a sudden it drops, it halves. So part 1, it says, describe the process that produces the change in the mass of DNA between W and X. So between this and this. So that process is called DNA replication. And DNA replication always takes place before any sort of cell division, whether it be mitosis or meiosis. So just to do a little bit of recap, I've included a diagram here. So during DNA replication, you're synthesizing a new strand or new strands of DNA through complementary base pairing, meaning that adenine or A pairs up with T and C pairs up with G, right? So the 2 strands would split first, and then the free nucleotides are able to pair up via complementary base pairing. And that causes the mass of DNA to double. DNA replication causes the mass of DNA to double during replication. The 2 strands separate and the newly synthesized strand is formed using, so always mention the term complementary base pairing. Complementary base pairing. Therefore, this doubles the original mass of DNA. Okay, let's go through the marking scheme. Number 1 is mentioning DNA replication. Number 2 is making the new strand through complementary base pairing, and that would double the original mass of DNA. All right, the next part 2. Describe what is happening in the cell between points Y and Z. So Y and Z are here, and you can see it drastically halves in the mass of DNA. So what's happening here is that the cell is dividing into 2, right? So that halves the mass of DNA, and that process is called cytokinesis. Cytokinesis occurs in which the cell splits into 2. All right, let's do part 3. The production of gametes involves the process of meiosis. On the grid, draw a line to show the changes in the mass of DNA during meiosis. So I've included a diagram here that showcases what meiosis looks like. So you can see here, the first step, you can see that the DNA doubles, and that's because DNA replication occurs. Right, so you're doubling the amount. And then what happens is, it swaps bits of the DNA with each other. And then afterwards, there is a cytokinesis event, right, where the mass of DNA would halve, and then another event in which it would halve again to form 4 daughter cells. All right, so the graph would look like this. So DNA replication would occur, so it doubles in mass. And then I'm just going to draw this from here to here. And then afterwards, the first round of cytokinesis occurs. So it halves the mass of DNA. So from 4 to 2, I'm just going to draw a straight line here. And then the second cytokinesis occurs, in which it also halves. So remember, this goes from 2 to 1. Right, and then we just draw this line here. All right, let's go on to question 5 in the next video. Let me know if you have any questions about this, and just leave them in the comments down there. All right, good luck. Bye.